Szanowni Państwo, dzień dobry. Good afternoon. Today, in our ECOTECH seminar, we've got a great pleasure to welcome Professor Stefan Franzen, who came to us all the way long from North Carolina. He's a professor of chemistry in North Carolina State University. Professor Franzen was graduated in chemistry in 1982, last century, as me, as I said, from University of California in Berkeley. And he got a PhD degree also in chemistry in Stanford University in Palo Alto. And today's lecture actually will be devoted to science itself in general, not to practical, some aspects in structural chemistry, biochemistry, spectroscopy, as Professor Franzen is expert of. And it will be devoted to science, as the title is Scientific Self-Deception. Are any of us immune? And Professor Franzen, please, the floor is yours.
Thank you for the kind introduction and for the opportunity to speak with you here today on a topic that it's not an easy topic for any of us to talk about. And I certainly do not talk about it with great joy, but I talk about it because of my own personal experience, which I will talk about a little bit at the end. But this is not about me, and this is not about any one of us as individuals. It's really about how we conduct science as a community. The thing we have to start to think about in a more serious way is, how do we evaluate science? How much time do we give young people to find their way in science? As we all know, there's a lot of pressure on people today to produce. That's the measure of success. That is what determines whether you proceed, whether you have a career, whether you have a salary, everything. Publications and all of the measures of success depend on what we do in the laboratory. Now, let me just start by saying that in general, when we talk about scientific ethics, we can begin by first defining what is the scientific method. What should we be doing? Well, we should make a hypothesis about nature, say, I think nature is behaving in this way, and then propose experimental tests to see if that hypothesis is correct. If it's correct, we can say it is corroborated. Can we say it's proven? I'll talk about that in a little bit later. What does it mean to be proven? The second thing that is very important to stress is that there is an implicit honor code in science, as in much of life, right? Science is no different than banking or so many other areas where honesty is essential and is assumed, really. So that's our honor code. We all accept that. But in my experience at Berkeley and Stanford and during my training, no one ever talked about that. It was just assumed that you're going to do the right thing. And if anyone did the wrong thing, it was assumed that it would be discovered almost immediately because of the way science works. People imagine, oh, another experiment will be done, and then it will be obvious that you lied. It actually doesn't work like that. It's not so simple. And ultimately, as I see the matter now, even the people who have committed what we call falsification or fabrication, in many times they don't even fully realize it themselves. In other words, they deceive themselves. So self-deception, which is the topic of this lecture, is the failure to employ the scientific method because one is so confident in one's own knowledge. one is so confident in one's belief that, oh yeah, I understand this system, I've got it, I know what's right. But in fact, for thousands of years, philosophers, and now today, I think psychologists as well as philosophers, have been concerned about a problem that we scientists should take more seriously. And just a side note is that many of my colleagues in chemistry and physics and other of the so-called hard sciences take a rather dim view of psychology. Some people even laugh, oh, it's not really a science, you know. And what they mean is you can't do the same kind of experiments that we can do in physics and chemistry. And that's true. You can't do the same kind of experiments when people are involved and when the human brain and its reaction to reality is involved. But that does not mean that we should dismiss it. That does not mean that we should look down on it. In fact, we really should respect it a lot more and really start thinking about how what we do is related to philosophy and is related to psychology. Does that mean we all have to get degrees in psychology? No. I think we just need to start thinking about this differently, not only in our educational system, but all the way up to the funding agencies and every single level in between where science is judged and evaluated by those who make decisions about how we're going to fund science and what we're going to do in science. So, thousands of years ago, the Greeks had two concepts that I think are really important for us to keep in mind. One I think is pretty well known, and that is hubris. Hubris is really pride, excessive pride, a kind of belief that you can do no wrong. And the second one is akrasia, which is really closer to what we are talking about when we talk about self-deception. It is the belief that my judgment is so superior that I really can do no wrong. It's related to hubris, but it really is like self-deception. And in fact, even Confucius began to talk about this topic, although the most comprehensive treatise started in the Song Dynasty in 1100. Zhu Si wrote the Zhongyong, which is the doctrine of the mean, and Złoty Środek, I think you say in Polish, which is a really fantastic and interesting book itself. But if you go back to Confucianism, Confucius and all the Neo-Confucians in the Song Dynasty and after, who wrote, you know, thousands and thousands of treatises, literally, on this subject, talked about extending knowledge, which is, of course, what we do today in science. And in fact, they had a concept that they called gewu, which is basically scientific analysis. It means to break things apart into its constituent parts and to understand it. But they also, at the same time, were writing in every single treatise on this subject about sincerity, which we could call honesty, I guess, chengyi, which is, for them, was extremely important. They were very aware that these two had to go hand in hand, that searching for knowledge and extending knowledge, the je concept, had to involve this sincerity and a search for the truth. And in fact, the first person in Chinese philosophy who really directly went after this was Zhu Si, who first wrote down the concept of ziqi, which is self-deception itself. And he wrestled with the question, is self-deception intentional? And this is a really, really difficult question because, I mean, what does it mean to say something is intentional? How do you prove intent? Was a false statement an accident, a mistake, or was it an intentional deception? Or what other possibilities are there, in the spectrum between these two? So now if we look at Western science, starting from the Greek tradition, we can actually see that there are examples of self-deception. I would call it self-deception because I do not believe that most of these scientists intended to do anything unethical. They didn't set out to say anything dishonest. But in the end, they did some things that today, if we examine them by our standards, we would consider falsifications or fabrications. And these include Ptolemy, who is maybe not, the most famous scientist, but of course he was for more than 1,000 years, actually 1,400 years until Copernicus. His view, we thought it was his view that he published about the solar system was the accepted view of the Catholic Church and many others. Turns out he stole it from a philosopher in Crete whose name we do not even know, but we know that the original documents were written in Greek and that they were found in Crete, and Ptolemy copied them. Then there's Isaac Newton. Isaac Newton, in his third Principia, he, as we say, fudged the numbers. He changed numbers so that everything would work out. For example, he calculated the speed of sound, which is impossible because to calculate the speed of sound, you need to know what heat capacity is. And Isaac Newton did not know what heat capacity was. It was not defined in his time, and he never defined it. He just made the numbers work out so that his answer was the answer that everyone knew was correct. And that's just one example. This has actually been studied by many, many people, even up to our time, because, of course, it's remarkable that probably the most famous physicist of all time actually committed what today we would have to call either falsification or fabrication. I don't know exactly which it is because it's a strange thing to do, to change the numbers so that everything works out just right. Turns out Charles Darwin was guilty of image manipulation. Gregor Mendel, he fudged his numbers or he changed his numbers so that the genetics would work out. Pasteur actually falsified an experiment by using his nemesis was Koch, the German scientist, and he took Koch's inoculation and used it in one of his experiments because his was not working. And he didn't tell anyone this. Now, and let me just finish by saying Millikan, who is credited with determining the charge on the electron, he cherry-picked his data, meaning he threw away about 1/3 of his data and only accepted the 2/3 of his data that gave him the answer he wanted. So here are some famous people. Millikan won the Nobel Prize. Pasteur, I mean, he was a great scientist. Mendel, he really is the founder of genetics, right? Darwin really did develop the theory of evolution. Newton really was one of the founders of modern physics, and so on, right? I mean, these are people who did great things for science. So the fact that we now can see the human weakness does not mean that we should think less of them. Some people think that we should somehow defend them and say, oh, they didn't do these things. Why? Why shouldn't we look at the reality and say, even some of the greatest scientists did things that we now can recognize as falsification or fabrication? Okay, so actually I'm just going to read this slide because this is kind of a complex thing to say. So there really is a classical ethical conflict that all scientists must confront. Science is impersonal. It doesn't matter who makes the discovery. If one scientist doesn't realize what's going to happen or what the result is going to be, what the next step is, someone else is going to do it. The inexorable logic of science just drives science forward. But for each of us in science, the credit for scientific discoveries, our own personal credit, is crucial for our career. So while science as a whole, it doesn't matter whether I discovered it or anyone else discovered something, but to me, it matters. So the credit for the discoveries is of pivotal importance for a person's career, for the individuals who are involved. And the competition is brutal. People are, constantly trying to be first and trying to make sure that everyone knows that they are first. And therefore, the temptation to deceive oneself, to think, oh, I've made a breakthrough, to claim a breakthrough, that temptation is enormous. And therefore, I maintain that breaking the honor code is much more common than anyone wants to admit. This is not to say that everyone deceives themselves, but rather that we all, and scientists and everyone really, has an internal struggle with the truth. And that is what we really have to confront, is that as individuals, we must begin to examine ourselves in a more careful way as we move forward. But we also need a scientific system that allows us to do that. Because if you're going to punish people because they take extra time to do extra experiments, because they are careful, then you're discouraging people from doing exactly what they need to do. And so how can we blame the scientists who are trying their best to do their best most of the time, and I mean 99.99% of the time, and yet in the end, about 1% of scientists actually end up cheating in some way that they get caught. So now let me start looking at this a little bit more quantitatively. Science is, of course, about data, and one of the ways that we characterize data is to look at the statistics of a result and decide whether a result is meaningful from a statistical point of view. This is usually done using a Gaussian function, shown here, the bell-shaped curve. The mean is, of course, the average. That's the peak of the curve. Sigma is the standard deviation, which is proportional to the width of the curve. And if we go out to 2 times sigma, the standard deviation, then the shaded area under the curve is about 95% of the area. Actually, it's, to be exact, it's 95.6%. But usually people are like round numbers, so people often just say it's 95%. Now, that is used, that model for computing data and the errors in data is used by everyone. It's absolutely universal. But now, Gaussian functions and the Gaussian statistic model has been used, particularly in biology, life sciences, and medicine, for another kind of evaluation, which is known as significance testing. So I want to explain that because in those sciences, this is a major problem for ethics. So this first was used in genetics by Ronald Fisher in 1926, and he used the Gaussian model to prove or disprove a hypothesis. So here's how he did that. First, he would define the average of some result. Then, he would define a statistical boundary, 2 sigma that I showed you in the previous slide. In other words, we could say if a number, a new measurement is made, there's a 95% probability it will be within a width of plus or minus 2 sigma of the original measurement. Third, if we want to find some alternative result, like for example, if we're testing a medication and we want to know if the medication will shrink a tumor, we measure the tumors in a series of patients, we find some mean and some distribution. Then we break the patients into 2 groups. One group receives the standard medication. That's the control group, we could say. The second group receives some new medication. And the question we ask is, are the tumors in the new group smaller than the tumors in the original group? Has the drug decreased the growth of the cancer? Which obviously is a desirable result in cancer therapy. And the criterion for saying that that has occurred is that the new result is outside of the 95% region. In other words, there's a 5% chance that the new result is simply a random result. If you're even farther outside, then it's less than 5%. Let me show this with a picture. But first, I just want to define something called the Null Hypothesis. That's really the control group I was talking about. And I did not come up with this nomenclature, and I really don't like it. I mean, why not call it the control hypothesis or the original hypothesis? That's much clearer, right? But everyone calls it in statistics, they call it the null hypothesis. That comes from Ronald Fisher. So here's the idea that I'm trying to describe. So there's some original group called the null hypothesis, or the control group. And if we're talking about cancer tumors, which is an easy example to talk about, although it's not something that's very nice to think about, there's some average size of tumors in a group of patients. There's some width that you can see here from this Gaussian. Does this work like this? Yeah, so some width like this. The 2 sigma line drawn here determines whether a medication or a new treatment will give you a statistically meaningful improvement. In this case, because the new mean, mu1 shown here, is outside of 2 sigma, it's further away than 2 sigma, we would say, oh yes, this is a statistically meaningful result. But here's the problem. Why did we say that the cutoff was 2 sigma? Why didn't we say it was 3 sigma or 4 sigma? It's completely arbitrary, actually, right? To say, well, this is what's statistically meaningful is just a matter of a society of scientists agreeing on something by convention. But there's nothing magical about it. There's nothing intrinsic or inherent about this number 2 sigma. But what's happened today is that it has become the dogma. If your result is outside of 2 sigma, you say it's statistically meaningful, and you declare that you have accomplished something, that your hypothesis has been proven. The drug works. We have a drug which can help cancer patients. But in fact, first, that's arbitrary because we chose 2 sigma as a completely arbitrary number. And secondly, it invites a really bad self-deception because many, many times the result is much closer to 2 sigma than this. And then the question becomes, is the new result really outside of 2 sigma? And now, probably when Ronald Fisher developed these ideas, he didn't think that this was the way it was going to be used. At least that's what most people think today. He was simply trying to define a method by which he could understand the genetics, that the alleles of genes that had been described by Gregor Mendel. And in fact, I'll come to that a little bit later because what he discovered really shocked him, and I'll tell you about that in a moment. But what he did was he came up with this procedure that I just described. So first of all, we start with a hypothesis. We call it the null hypothesis. It's the starting hypothesis or the original hypothesis. Then we test it by collecting data. Then we determine whether the events in the data support or negate the hypothesis. And we reject the null hypothesis. In other words, we say that an effect is real and has really been achieved if the result is outside of the 95% limit. In other words, if there's less than a 5% chance by just random error that we achieve that result. But in fact, Fisher never said it had to be 5%. That came later. Fisher just said there's a cutoff somewhere, and if the result is outside that cutoff, then it is statistically meaningful. There's actually another kind of statistics that is now gaining popularity, and I think really many people are saying we should move to. It is called Bayesian statistics. Bayesian statistics is based on degrees of belief. And the idea is I make a hypothesis. It's called a prior. I conduct experiments. So this sounds very similar. And then I use what's called a joint probability, which according to Bayes' theorem basically says if the evidence combined with the original hypothesis moves me in a direction that increases the probability that the hypothesis is incorrect, then I will say that I'm moving in a good direction. There's no absolute cutoff in Bayesian statistics. There's no magical line like there is in the interpretation of Fisher's idea, what we call significance testing. So the posterior, the result that you obtain afterwards is either better or worse. And now comes the question, how much better? How much worse? well, then we can discuss it. But no, there's no yes or no, all or nothing answer the way there is in significance testing. So we have Bayes and Fisher, these two ways of looking at statistics, which of course, statistics are essential in almost all of science when we do experiments to be able to say whether an experimental result is meaningful and meaningfully different from a starting point. So I'm going to, just in the interest of time, leave it there because it's a very complex issue, that is to say, Gaussian versus Bayesian statistics. But what it gives you, hopefully, a sense for is where the dilemma can come. So the dilemma can come in how we evaluate our results. If we have an all or nothing criterion, then there's a tremendous pressure to get a value that is greater than, or that is outside of this 2 sigma width. And actually that's described by something called the p-value. That's the probability that the result would occur by random chance. And since the arbitrary cutoff has been set at 95% of the area, that is to say 5% of the area is outside of that width. If the new result is outside that width, then the p-value has to be less than 5% or less than 0.05. So, ethics of science is determined by how we substantiate observations, what controls we do, what checks, what kinds of self-criticism, etc. I haven't even really gotten into that yet, but then it's determined by how we report the method, how honest we are, how complete we are. Then it is determined by whether our evidence is valid in a statistical sense, and that's what I've been talking about mostly up till now. And of course, how we document it, how we report it to others, how we respond to criticism, and how we question our own motives and prejudices. And this last point is the point that I think by the end of this talk I hope to come back to and really point out that this is where we need to head. We need to instill in each student who comes into the scientific world a sense that we can understand our own weakness by studying what other people have done and thereby prevent ourselves from making the kinds of mistakes that I have been describing here. This is all very much connected to the philosophy of science, by the way. In the 20th century, the most famous for many years was Karl Popper, who came up with the idea that theories can be proven wrong, but they can't be proven right. And there's some sense to that. I mean, you can say, okay, how would you prove something is right unequivocally? I can certainly prove something is wrong by showing a counterexample and saying, oh, there's an example that shows that's wrong. But how do I prove something is right? Yet, that's not very satisfactory because, after all, we want some kind of criterion for acceptance, and Popper doesn't give us that. Then came Thomas Kuhn, who pointed out that one theory replaces another theory, not in a gradual sense, but in a sudden sense. There are what he called scientific revolutions, and I've outlined some of them here. The Copernican Revolution, of course, changed our view of the solar system, and it was radical, right? We moved from an Earth-centered to a sun-centered solar system. Then Newtonian physics came along and changed our whole view of how motion occurs in heavenly bodies and here on Earth as well. Then came the quantum revolution, in which we realized there's a whole different set of rules that applies when we get down to the level of molecules and electrons and nuclei and so on. And finally, there's been the genetic revolution, in which we began to understand that biology is something that we can actually manipulate, something that we can actually control. So, while in one sense it's tempting to say that these are revolutions, you can also look at science as an evolution. At each one of these steps, people had to make experiments, come to the conclusion that something was not working, and then move forward in a small incremental way. So, in some sense, the Kuhnian idea of revolutions is an oversimplification. Then along came Feyerabend, who basically said, there is no one scientific method, all implementation is subjective. And finally, the deconstructionist, who said science is not unique in its view of the truth. They believed in cultural relativism. So the fundamental question is, how do we define valid evidence? Let's start with cultural relativism, because I want to dispense with it. I don't believe that the deconstructionists understand science, and neither did Alan Sokal, who wrote the article, and I love this title. The article was titled, "Transgressing the Boundaries Toward a Transformative Hermeneutics of Quantum Gravity." If that sounds like nonsense, it's because it is. This article is complete gibberish. He got it published in the journal Social Text, and his point was that the people who are criticizing science from a subjective point of view and saying everything is relative, you know, that a philosophical view from India in 2000 BC is just as valid as quantum physics for describing an electron. Well, that's ridiculous, right? Because in 2000 BC, no one knew what an electron was. So his point is that science has a set of rules. We have to look at those. We have to build on those. If we throw that out, we've thrown out everything. So that's kind of a simple one to deal with. But now we have to move to the more serious question, which is how deeply entrenched is this problem that I've been describing? And well, here's a book that really shocked me when I learned about it. This book was written in 1830, as it's kind of hard to see because I couldn't find a good high quality copy of the title page. The book was entitled Reflections on the Decline of Science in England and Some of Its Causes. In 1830, the decline of science in England, most of us would think that that's when science was on the upswing, right? Everything was being discovered, etc. So why did he write that? Well, he was actually very disturbed by Sir Isaac Newton, for one thing. So he already knew in 1830 that Isaac Newton had manipulated the numbers in his Principiae. But in fact, there were a lot of other scientists who he pointed out had published falsified data. And he could show that, and he did in his book. Now, in more modern times, there have been hoaxes that have led to self-deceptions, like the Piltdown Man, which was a supposedly humanoid that was discovered in Great Britain in Piltdown in 1912, and it was a hoax. And yet, almost everyone seemed to believe it. The hoax lasted for 40 years. Here's a picture of the Piltdown Man skull. It was an orangutan skull that was, or actually it was a human skull with an orangutan jaw put onto it, and then some other changes made to it. And why would anyone believe this? Well, the theory that I have read is that it fed into a national sense of pride in Britain. Britain is the cradle of civilization. This is where humans started. Sounds good, doesn't it? Or N-rays. N-rays were discovered in 1899, I think, by Henri Blondlot. No, I'm sorry, 1905. René Blondlot. And he claimed that there was a new kind of ray, like X-rays, only they were called N-rays. And he had a detector and he had everything set up. And famously, an American scientist named Dr. Wood traveled to Nancy, where Blondlot lived, and he went into the dark room with Blondlot, and while Blondlot was showing him the N-rays, Wood put something in the beam, supposed beam, that was being studied. He blocked it. But Blondlot could still see it. Obviously, he was imagining it. Blanc law never accepted it, and many, many French scientists never accepted it. They saw Wood as a troublemaker. You know, what's this American coming to France to try to show us how science is done? We know how science is done. It was a sense of national pride. So in talking about these kinds of things, very famous scientist Irving Langmuir said the following: the characteristics of N-rays and other pathological examples are cases where no dishonesty is involved, but people are tricked into false results by a lack of understanding about what human beings can do to themselves in the way of being led astray by subjective effects, wishful thinking, or threshold interactions. Well, he's describing self-deception, obviously. He's saying, you know, these people deceive themselves. And the point to notice here is he implicitly is saying that there is no intent. And this exactly was the question that Jussieu wrestled with almost 1,000 years earlier. What exactly is intent? How do we define intent? That's a real problem. Why not just admit the mistake? Why couldn't Henri Blanc, when he learned that somebody had blocked the beam, just say, oh, you know what? I've been imagining this the whole time. Why couldn't he do that? Is that just a self-deception? If someone proves to you that your self-deception is a self-deception and you still persist, now can we say there's no intent? So when does it become intentional? And when does it become unethical? These are the real difficult questions. There are just so many examples, and I'm not even going to try. I mean, I could literally give a 3-hour-long talk of examples, so I'm not going to do that. But one very famous example of a self-deception, both of scientists themselves and of the general public, is Laetrile. Particularly when I was young, everyone was talking about Laetrile as a cure for cancer. And there were people who would travel to Mexico to get Laetrile because you couldn't buy it in the United States. It was illegal. The Food and Drug Administration outlawed it. And it has been studied so many times. And every single study has conclusively shown that Laetrile has no effect compared to the null hypothesis. You just get the same result as the null hypothesis. It does nothing. And yet, even today, it keeps resurfacing. People keep talking about Laetrile, not the way they did in the 60s, but it's still out there. Or one that we all know about today, vaccines, right? Why are people so convinced, some people, that vaccines are harmful? Well, it's interesting. This started actually in 1998 when Andrew Wakefield published a study based on a study of 12 children. That's a pretty small sample. And he purported that there was a connection between vaccines and autism. And he did this because he was a witness he was an expert witness in a lawsuit by some parents whose children had autism, and they wanted to blame the autism of their children on a vaccine. So he did this study, which it turns out was completely bogus and falsified. But he did testify at the trial, and ultimately he was discredited and he lost his medical license, and you would think that would be the end of it, wouldn't you? But no, then people in Hollywood started to jump on this. You know, vaccines are harmful, vaccines cause autism, and many, many thousands, tens of thousands of people refused to vaccinate their children, particularly against measles. And as you may know, there have been measles outbreaks now. Measles was eradicated. It was gone. Now, in California and southern Britain, there are outbreaks of measles that affect dozens, hundreds of children, and children die. And of course, as we know, today, with the problem of COVID, this is something that is just continuing to grow. It has become almost a mania with many people. But in fact, you can trace it back to a scientific deception. And I guess in the case of Andrew Wakefield, I would say it wasn't self-deception. He knew what he was doing. But then so many people following him deceived themselves. So let me talk about a case study that we're all quite familiar with today, and that is climate change. But I want to go back in history a little bit. I want to talk about the first real description of the problem of burning coal or other hydrocarbons and producing carbon dioxide. This was recognized first by Svante Arrhenius, the Swedish physicist, in 1896 in an article that he published. But very quickly, his nemesis, Knut Ångström, published an article in 1900, and I'll show you that article in a moment, to question Arrhenius's finding. Now, Knut Ångström, I must observe, is the son of the famous scientist Jonas Ångström, and Ångström being the unit of length that's used most often in chemistry is a name that many people know. It comes from Jonas Ångström, the father, not the son. Knut Ångström was nothing like his father. I don't want to say whether he was a good or bad scientist. I don't really know, but I'll show you that what he did was something less than ethical. So Ångström's article that I'll show you stands out even today, not only as a poorly designed experiment, but really a self-deception. So, of course, there are reasons for these self-deceptions, and that's why I want to go into this case study. One reason that's quite obvious is that from the very beginning, if you admit that burning coal even produces carbon dioxide, and that carbon dioxide in the atmosphere will cause some kind of heating effect at the Earth's surface, well, then you have to start looking for alternatives to coal. So there's an economic reason not to want this to be true, and a very, very powerful economic reason. But secondly, Ångström was Arrhenius's rival for the physics chair in Uppsala, Sweden. And so Ångström wanted to make sure that he would get the chair. He probably would have anyway because he was Jonas Ångström's son, but he wanted to prove that he was right and that Arrhenius was wrong. And these really are two factors that we see over and over again, reasons for self-deceptions. Number one, economics. You know, if whether you're looking in the environmental chemistry area, medicine, or anywhere else, a finding that is economically painful is probably going to cause the scientist who made that discovery some pain. And so many times scientists just don't want to discover these things. Or if they do discover them, others will want to prove them wrong at any cost, because there will be a lot of money behind the effort to prove that there's no lead in the drinking water, or to prove that, you know, global warming is not occurring, or whatever it is that you want to prove. And the second is, of course, the competition for not just fame. Fame is, of course, something it's hard to even put our finger on, but there's the competition for jobs, there's the competition for grants, there's the competition of every kind in science, and that drives a lot of people's behavior, which is why, as I started out saying, we should not discard the psychologists. We should really incorporate a certain amount of psychology into our thinking about what we do as scientists of any kind. Now, a lot of equations here, but I just want to point out that the simple idea that Arrhenius pointed out was that, of course, the power that's coming to the Earth from the sun must equal the power that's being radiated by the Earth. All heavenly bodies radiate thermal radiation. It's called black body radiation, a name which I very much dislike because it confuses students to no end. It's really thermal radiation, right? The sun has a temperature, and so there's a flux, a radiation flux that depends on this temperature, and the Earth then absorbs and re-radiates, and that's what determines the temperature of the Earth. And in fact, if you take all these equations here and you write them in a very, very compact form, you can calculate the temperature of the Earth from the temperature of the sun by knowing the radius of the sun, in other words, its surface area, by knowing the radius of the Earth's orbit around the sun, and the reflection of light at the top of the atmosphere, called the albedo. And those are the only things you need to know. Using those, we can accurately calculate that the temperature of the Earth is 255 Kelvin. Ah, wait a minute. It isn't 255 Kelvin, is it? That would be the truth if that would be the case if the Earth were just a rock, had no atmosphere. Then our temperature, surface temperature, would be 255 Kelvin, and there would be no life on Earth. The reason we have life is because of the atmosphere, because of the greenhouse gas effect. And this has been known actually since 1880. So how does that work? Well, again, we have to have this equilibrium. The flux in has to equal the flux out, or I should say the power in has to equal the power out. But when we calculate it at the top of the Earth's atmosphere or from the surface of the bare Earth, the solar flux coming in has to equal the flux going out. Now, the solar flux in an Earth with an atmosphere has to equal the flux going out from the top of the atmosphere. But we don't live at the top of the atmosphere, do we? We live on the surface of the Earth, and so that means that there's going to be more radiation from the surface because there are going to be losses in the atmosphere before the radiation reaches the top of the atmosphere. Now, there are some simple facts that we know today that they didn't know in 1900, like, for example, that the amount of carbon dioxide is increasing dramatically in the atmosphere, and there's really no argument about that. We also know today that the temperature of the Earth is increasing, and there's some argument, but there's not much argument about that either. The real argument is about the correlation between the two. Here's a correlation. The correlation between the letters in the winning word of the Scripps National Spelling Bee and the number of people killed by venomous spiders. Well, obviously, none of us believe that that correlation has any significance, but the correlation that I just showed you between the amount of carbon dioxide and the temperature is a lot more significant than that. There's causality between them, and causality requires a hypothesis. So the hypothesis we can make is that the global temperature rise in the past 100 years has been caused by an increase in CO2 in the atmosphere. Okay. Again, not easy to argue against that in certain ways, but we need a theory. We need some way to test the hypothesis. Well, we can measure the absorption by carbon dioxide in the laboratory, and I'll show you that Ångström did that, or he thought he did it. And what it is that causes this absorption is the bending mode of carbon dioxide. The bending mode of carbon dioxide is not exactly at the peak of the Earth's emission spectrum, but it's not far. And so it can absorb the Earth's radiation, of course, and cause warming. Then we need some way to validate the theory, to test the evidence. So we can measure the intensity of that vibration, and we can determine the optical path length of the atmosphere by a calculation, but that's pretty solid because we know what the pressure is, we know what the densities are, etc. We can calculate that accurately. And then we can use the theory of thermal equilibrium that I talked about earlier to determine the constraint on the flux at the top of the atmosphere. So far, so good. But now, if we want to say the following, now's where the argument's really going to happen. If we say the increase in CO2 in the atmosphere in the past 100 years has been caused by human combustion of fossil fuels. Now a lot of people, not a lot of people, but enough people that it's significant, will start arguing. No. The CO2 is coming from volcanoes. The CO2 is coming from the ocean. The CO2 is coming from somewhere else. It's not us. We couldn't possibly burn that much, they will say. And yet, we can also show that we know exactly how much petroleum we're pumping out of the ground and burning, how much coal we're burning. We can calculate all that. It's not hard to look up what those numbers are and figure out that we're pumping 28 billion tons of carbon dioxide into the atmosphere each year. And then you can start to ask, I rounded up to 30 billion. It's probably 30 billion by now anyway. That's an old number, 28 billion. So there is a lot of evidence, actually, that humans are causing this, despite the fact that there are those naysayers, those who don't believe it. Well, are they engaging in a self-deception? Let's look at the first self-deception. So first of all, you know, what I'm going to maintain is that the people today in think tanks and those few university experts, of course, there are a minority of people in universities who would say this, who would say that the correlation is impossible because, for example, they might say CO2 absorption is saturated. There's already so much absorption by CO2, adding more won't make any difference. That's an argument that people make. Or, like I say, they'll say that the source of the CO2 is something else. But this saturation idea is a really big one with the people who want to argue that that none of this has to do with humans, or that there's nothing we can do about it, even if it does have to do with humans. So Arrhenius predicted all of this in 1896, and yet by 1900, no one believed him. Why? Well, sometimes the acceptance of a scientific idea doesn't depend on the facts, doesn't depend on the experiments, but it depends on who says it. Sadly, we see this all the time. Oh, so-and-so is a good scientist. We believe the work that comes out of that person's lab. So-and-so, maybe not. It's who says it that, not always, of course, but in many cases, the person who's believed is believed because of their reputation, because of some perception that they do good science. And of course, there are these other blinders that we have, the economics and all the things that I've been talking about. So let's look at Knut Ångström's paper. Now, I'm not going to assume that you all know German, but this is just for fun, just to look at what the article looks like from 1900. And here's my translation. On the importance of water vapor and carbonic acid, of course, that's his word for carbon dioxide, and carbonic acid absorption in the Earth's atmosphere. And he writes, the importance of water vapor for the absorption of the solar and terrestrial radiation is already well understood based on the work by Tyndall. On the other hand, the views of the magnitude and intensity of this absorption are still contradictory. The effect of carbonic acid is less well understood. Okay, so what did he do to try to understand the effect of CO2? So he describes how he went on a trip. Yeah, that's how this has to start. We have to go on a trip. He went on a trip to Tenerife. I kind of wish I could go to Tenerife too. And he climbed up Pico de Teide. I'll show you, actually, I'll come back to this in a moment. Here's Pico de Teide. I love this picture. He climbed up to the top of Pico de Teide to make this measurement. And what he did, he carried this box up to the top of Pico de Teide. It had two tubes in it. One tube was filled with air at 1 atmosphere, and one tube was filled with carbon dioxide at 1 atmosphere. And he pointed these at the sun, and he measured whether there was any difference between the air and the carbon dioxide. And he concluded that there isn't, that the carbon dioxide isn't absorbing anything significant, and therefore, there's no problem. But wait a minute. And I actually don't have time to go through all of this, but I explain kind of what he did. But if anyone is interested, these slides would be available for anyone who wants them. I will leave them here. They're you know, a public document. But it's not the Earth's, it's not the sun's atmosphere. I'm sorry. It's not the sun's radiation that matters. It's the Earth's radiation that matters, right? The Earth is emitting radiation at about 15 microns, and actually about 10 microns, I'm sorry. It's absorbed by carbon dioxide at 15 microns, its peak. And it's the absorption of the upward going radiation that's causing the warming effect, not the solar radiation being absorbed as it comes down. Actually, if there were absorption of solar radiation coming down, it would not cause a warming effect, it would cause a cooling effect. right? If less radiation reached the surface, that should cool the Earth. So here's the picture from his paper, from Ångström's paper. So this is the so-called black body curve, the thermal radiation curve. Here are the carbon dioxide bands. Here's the bending mode. Here's the asymmetric stretching mode that everyone agrees is not so important. And he chose to do an experiment at 373 Kelvin, 100 degrees centigrade. I still don't really understand why. And he says that 10% of the radiation was absorbed in a length of 30 centimeters at a pressure of 780 millimeters, which is very close to 1 atmosphere. Why did he choose 30 centimeters length? Why did he choose this? He was trying to simulate the actual absorption by the atmosphere. And it turns out his numbers were off by a factor of 15. Then he reduced the pressure by 2/3. So he went from 780 to about 1/3 of an atmosphere. And he says that the change was unbedeutend. That means, of course, tiny, minuscule. So this tiny change of 0.4% occurred in the total radiation. Now, if you go through the numbers and you analyze that, it turns out that that number is 15 times smaller than the actual number should be. So all of his numbers are off by a factor of 15. What's the point? The point is, when you go through the analysis of his data, aside from some mysterious factor of 15, his data show exactly what is true, which is that carbon dioxide absorbs the radiation coming from the Earth. And if we increase the amount of carbon dioxide, well, he decreased it, so he was looking for an increase in the radiation coming out. But if we increase the CO2, there'll be a decrease in the radiation that's leaving the Earth. And here's a picture from actually a paper that I wrote, actually with my father, who became very interested in these questions, which is why I learned so much about them, because I wanted to work with him. He's in his 80s, and this is his occupation these days is to think about what's going on with our atmosphere and how can we convince people that this is an important problem. So these are some calculations where what we did was we used the publicly available HYTRAN database to calculate what the spectrum of carbon dioxide looks like, and then to put that spectrum on the black body curve of the Earth, not at 373 Kelvin, but of course at about 200 and whatever it was, 287 Kelvin, I believe, which is considered to be the actual temperature of the Earth. And this is just a sample calculation to show you that this is not saturated. If we start increasing, now these are huge increases just to make the point, right? But if we increase the pressure of carbon dioxide, the absorption still has plenty of increase possible. It's not saturated. The people who say it's saturated have simply not done the calculation and not looked at this. This is all publicly available data. There's nothing hard about it. So here's what I'm trying to say is the way this works. For the bare Earth, the radiation out equals the radiation in. If we now put an atmosphere on the bare Earth, that will absorb some of the radiation. Now the radiation out is less than the radiation in. That's because of the transmissivity of carbon dioxide. So in order to reestablish radiation equilibrium, the radiation from the Earth must increase. In other words, the Earth must heat up. And when it heats up, then after the absorption by the CO2, the radiation out will equal the radiation in. So here, bare Earth, they're equal. Here they're equal, but the temperature of the surface of the Earth depends on how much carbon dioxide there is in the atmosphere. And in fact, you can derive a very simple formula, and this is it. The temperature of the Earth with an atmosphere, so this really means the temperature of the Earth with an atmosphere, not just the atmosphere itself, is equal to the temperature of the bare Earth divided by the fourth root of the transmissivity of the atmosphere, which is mostly carbon dioxide absorption. So due to carbon dioxide absorption. So the point is, oh, and let me just mention that, of course, all I used to obtain this result was the very well-known Stefan Boltzmann law that says that the radiation flux is equal to sigma times T to the fourth. These are the only facts you need: the HYTRAN database, the Planck law, the Stefan Boltzmann law, to prove, in a sense that I think a physical chemist or physicist would accept, that the temperature of the Earth depends on the absorbance of radiation by the atmosphere. It's not that hard, is it? And yet, people don't want to accept it. Ångström did not want to accept it. But what's even more disturbing is that the entire scientific community sided with Ångström. Arrhenius was forgotten for almost 40 years. And even 40 years later, when the scientists in the US Air Force began studying the upper atmosphere and started to realize this connection, they were interested in other things than atmospheric change, but they realized the connection. Their work was soon dismissed as well. It wasn't really until the 1970s that people began to start thinking seriously about this problem. And as we know, even today, there are so many people who have a megaphone and who tell people it's all a lie, it's all not true, etc. And a lot of people listen to that. It is a self-deception. But one question that still plagues us is, what about this matter of intent? Did Ångström really intend to deceive people? Was he so interested in getting ahead that he somehow fudged his numbers? Why was he off by a factor of 15 in something so simple? He was a smart guy, that's for sure. So this is really perhaps the biggest problem. Now, I added these things, but I think I'm going to skip it. These are just some of the details of, how you can calculate the spectrum of CO2, the fact that there is a region that will be saturated, and then there are the wings, and so on. This is how we did the calculation. We first calculated the transmissivity, and then we multiplied that by the Planck curve, etc. So those are the details. Then we integrated it, and we finally, with that simple model, what do we predict? We predict almost exactly what the temperature of the surface of the Earth is today, and we predict almost exactly what the annual temperature change should be. According to most simulations, it's about 0.012 degrees per year. We got 0.014, and after about 100 years, we're predicting a temperature rise of 1.2 degrees centigrade. If you look at Hansen and some of the other really big-name climate scientists, they'll put a number something like 1.4 to 1.6. So this simple model, I'm not claiming that this model is in any way highly accurate. I'm just saying it's something that we can all look at and say, okay, there's a real issue here, and something that we should not be deceiving ourselves about. And yet, what if it is all a big hoax and we create a better world for nothing? Ha ha, would that be so bad? Okay, I don't know how much time I have. I don't know if I should go through this whole case of Mendelian genetics, but at the risk, let me just try here. So, of course, Mendel is famous for his experiments with peas, with pea plants. And he studied 7 different genes, and he discovered the idea of dominant and recessive alleles. And he noted that if a pea plant that has one dominant and one recessive allele is crossed with another pea plant that has one dominant and one recessive allele, that you get 75% dominant and 25% recessive. Okay, we all accept today that that's at a very crude level, an accurate way to think about alleles in genetics. But it turns out that Mendel's numbers were too perfect. In any real experiment, there should be some experimental error. If I grew 1,000 pea plants and I collected them and then analyzed them, would I get exactly 750 who had the dominant and exactly 250 who had the recessive allele? Probably not. There would be some kind of experimental error. But what's worse is that in reality, the 7 different genes are not completely independent. And we now know, and Ronald Fisher knew in 1926, that what Mendel had written back in the 1880s, I think, that was not possible from a statistical point of view. And it's a pretty long argument, so I'm not going to go through it. But it caused Fisher great concern Because, of course, Mendel by that time was accepted as the father of genetics, and he was revered by many people. No one wanted to believe that Mendel had falsified data. It sounds terrible, doesn't it? But as I've said all along, right, Mendel probably did this in all innocence, in a sense. He may have been convinced that it had to be true in some way. After all, Mendel was not what we would today call a scientist, right? He was a monk. He lived in a monastery. He was doing this out of his own curiosity and his own interest. He may have believed that for some reason things had to be a certain way. We'll never know. But what Fisher thought was that Mendel must have had a scribe, someone who wrote down his data. And it was the scribe who did it, not Mendel. That's what Fisher wrote. Mendel didn't do this, his scribe did it. Later on, two scientists in the United States wrote several articles and even a book to try to explain why Mendel was not guilty of falsification of data, as we would view it today. And the point of bringing this up is, does it really make a difference to us? The fact is that Mendel lived 130 years ago, or he did his work 130 years ago, and whether he did this or not, he still made a breakthrough in genetics. I suppose you could say what does matter is his intent, right? Did he intend to deceive anyone? I'm convinced he didn't. I'm convinced that he did this in a sense as a self-deception, in all innocence, in a truly non-intentional, self-deceptive way. To me, personally, that's excusable. We can understand that, particularly in the historical context. We don't need to write books to excuse him, because what that does is it starts to excuse other things. These people who wrote these books, in fact, one of them, this fellow Franklin, at the University of Colorado, also wrote things about me, claiming that I was someone who hated science and that I had tried to destroy two scientists. And I'll come to that in a moment. So, one last point about this whole analysis that Ronald Fisher came up with that then became significance testing and so on. So just to remind you, in significance testing, if a result is outside of this width of 2 sigma, then it's outside of the 5% window that we consider acceptable. In other words, we say that the p-value is less than 0.05. That means the probability of this happening by random chance is less than 0.05. Okay. If you do actually, I didn't do, I should put the reference down here, but people have studied studies. They've asked how many times do people get numbers that are bigger than 0.05, and how many times do they get numbers that are smaller? And a study of about 100 papers found that the average value was 0.043, which is kind of strange because that's close enough to 0.05 that it makes you suspicious that what's happening is people are doing what's called p-hacking. P-hacking is when you do your experiment with your initial hypothesis, you get your p-value, and your p-value is 0.051. Well, that's not good. That would say that your null hypothesis is correct, and that whatever you were trying to establish about a medication or anything else is not correct. And if you just had $1 million grant, and that was your p-value, you would feel pretty bad. But maybe, if you look at your data a different way, you can find a different p-value. Supposing you change which variables you will accept in your correlation. After all, in many of these experiments, there are a number of different variables. I've heard one example being jelly beans. So, supposing somebody says, jelly beans cause cancer. So, they study 100 people, and they find that if 50 people eat jelly beans, and 50 people don't eat jelly beans, the rate of cancer is the same. Well, then that basically means that the p-value is 1, and that means that there's absolutely no correlation. But then you start looking at whether red jelly beans maybe give you a correlation, or green jelly beans, or yellow, or some other color. And you discover that, oh my gosh, just if we look at purple jelly beans, P is 0.049. Hooray, purple jelly beans cause cancer, which is of course ludicrous. Because if you look at enough different possible correlations, just by random chance, one of them will probably have a p-value that is less than 0.05. Because again, this is all about statistics. It's all about chance. So when we see that the p-value of over 100 studies averages to 0.43, it has to make us wonder whether people are deceiving themselves. I love this picture. Oh, so you found a p-value less than 0.05. Please tell me about your breakthrough. We really have to think about what we're doing when we start using statistics. And this is just one article here about p-hacking. So, in fact, p-hacking is so prevalent. There have been now many, many articles written on it. Among the most famous is John Ioannidis at Stanford University, who published the article, Why Most Published Research Findings Are False. That's a pretty provocative title. And I'm not sure that I totally buy his argument. But in fact, there are so many ways that people can deceive themselves that we really have to be concerned about the quality of science, particularly when people are publishing 1 million papers a year. And of course, the estimates of the increase in fraud in science show that fraud is increasing, whether it's just error, plagiarism, duplication, or outright fraud, it's increasing. Of course, the number of papers is increasing. So that doesn't necessarily mean that the rate of people falsifying is increasing, but it's still an alarming trend, isn't it? Now, in the final few minutes, I just want to talk a little bit about my own personal experience and why I care about this matter so deeply and why that fellow Franklin in Colorado thought I was such a terrible person. When I was a young professor, about, well, 15 years ago, not that young, I guess, one of my colleagues who I worked with very closely was an author on this paper, published in Science magazine. And the claim that these people made was that they could create hexagonal-shaped palladium particles. And notice the scale bar here of 2 microns, because this is important. And what they claimed was that they could do this using ribonucleic acid, RNA. And if you make a sequence of, say, 80 nucleotides of RNA, and you use a procedure for randomizing that sequence, which basically you make a DNA template, and then in the middle you simply put in any arbitrary nucleotide so that you get about 40 nucleotides that are random. So that's 4 to the 40th power possible combinations. It's a huge number. It's something in excess of 10 to the 14th. So you have a library, as they call it, of different sequences. And so their idea was they would find a sequence, they colored it green, that makes this particle, and then they would centrifuge the particles, collect them, get rid of the inactive sequences, collect this active sequence, and then amplify it, and then repeat. So this is what they call their evolutionary cycle. And my dean came to me right after this was written and asked me if I would join them and form a center called the Center for RNA-Mediated Evolutionary Materials. So why me? Why did he come to me? Well, he said, they are not physical scientists, which is true. They need a physical scientist if they're going to get this grant funded. This was by the W.M. Keck Foundation, and it's one of those grants that's one per institution. So our college was competing with other colleges. We had to look good. Otherwise, we wouldn't even get off campus. Turns out, what I didn't know was that another one of these, and let me not name names, I mean, their names are public, but just not who said what or did what, but one of the investigators had a special friend on the board of the W.M. Keck Foundation. And this person knew that if we could just get this grant to the Keck Foundation, that we would get the money. Well, I didn't know this. I don't even know, my dean probably didn't know it either. But my dean had a very strong sense that we were going to be successful. He essentially told me that. So I don't know what he knew, but he said, please join them. Another thing I should mention is that I had worked with one of these people for a number of years before, although by this time I had basically begun to stop that collaboration, not because I suspected that he was doing anything wrong, but just because he was working on nanoparticles and I decided to try it for a while and then I lost interest in it, and because I'm really a biophysicist and it wasn't really what I wanted to do. Plus I met a plant virologist and I began working with plant viruses as another kind of nanoparticle, and that I did for 10 years, and so I was moving away from this. Yet my dean convinced me to join them. And so I did. And guess what? We got $1 million. And during this time, I also became very curious about the structure of this RNA, right? If the RNA is doing this, if it's really making a palladium particle, then it's got to have a structure, right? This isn't happening by something random in solution. It's acting like an enzyme in some way, isn't it? That's what they actually called it later. They called it a Palladium Ace, as ASE, as in an enzyme that makes palladium. How were they saying they made palladium? They were saying that they took this molecule, so here are 2 palladium atoms. Here's a carbon cage around those palladium atoms. This is called palladium DBA. It's actually a very commonly used molecule in organic chemistry for delivering palladium 0, metallic palladium atoms, to a reaction for catalysis purposes. But no one in organic chemistry would ever use this molecule in water or aqueous solution because this molecule does not dissolve in water. Only when they described this experiment, they did not say that they used a non-aqueous solvent. They said they used aqueous buffer, water. And being a physical chemist, I had never used palladium DBA, so I didn't look up the material safety data sheet. I didn't check it out, which I should have. I assumed that what they wrote was true, like we do in science, right? You read a paper, if someone says they did something, you believe they did it. I trusted that they had done what they said. But what they said was simply impossible because obviously RNA has to be in an aqueous buffer. In fact, if you even add 20% ethanol to RNA, it will start to denature. If you add 50% ethanol, it will simply fold up and fall out of solution, precipitate. And yet this molecule will precipitate in water. So there's a fundamental contradiction here. Okay, I'm not going to read that. Now, they got even fancier. In 2008, they claimed that they could make arrays on a surface and that they could form these palladium molecules with surface-attached RNA. Really fancy stuff. Here's the picture from their paper. Notice the scale bar here. This is very similar to that picture in Science, isn't it? But it's now 250 nanometers. What happened? Well, if the particles had been 2 microns big, they would have been too big for these arrays that were made by their collaborator at Lawrence Berkeley Laboratory. So this and other facts were things that alerted me to the fact that there was something very wrong here. And of course, I began asking the students, and I discovered that they were not using aqueous solution at all. And at first, they didn't want to tell me how much non-aqueous solvent they were using. So they were using tetrahydrofuran and water. Tetrahydrofuran, in many ways, is like ethanol in its properties. It mixes with water, it's clear, and it can dissolve molecules that water can't dissolve. But how much of this tetrahydrofuran were they using? First, they said 5%. Well, that doesn't sound like much. But then we began exploring that, and we discovered that 5% can't be right. You can't even dissolve this molecule in 5%. No. So I went back to them, and they said, well, actually, no, we usually use 15%. We tried 15%. No. Turns out they were using 50%. If they had written that in the science paper, that they used 50% of an organic solvent and RNA, no one would have ever published the paper. In fact, they would have been laughed out of the room. So they lied. Did they know they were lying? Or did they deceive themselves? Well, I began to ask them lots of questions. They started to threaten me with a lawsuit. They threatened the university with a lawsuit, et cetera, et cetera. And finally, I got very tired of this because I had a paper written that I wanted them to publish with me that showed I didn't ask them to retract their paper. I just wanted to publish what we found, and then I was going to resign from the whole thing. However, my university, if you can believe this, prohibited me from submitting the paper. And they told me the only way that I would ever be able to publish this paper is if it was approved by the research integrity officer. So I went to go see the research integrity officer, who immediately told me that what I had described was a case of falsification and that I should file allegations. It took me a while, but I finally did, because I had been threatened and my university had been threatened. And after all, when a university tells you you can't publish, there's clearly something wrong. But I had never seen the laboratory notebooks. I only knew what the students had told me, what was published. I knew there was something wrong. And then I started to ask for the laboratory notebooks. And the university took all of them and locked them in a room and said, no, you can't see them. These are evidence now. I said, well, yeah, they're evidence, but I mean, I'm the one who made the allegation. Don't I get to see them? Nope, nope, you can't see them. Finally, in 2010, I hired a lawyer, and I spent exactly $1,035 to get the university to release these notebooks as public records, which sounds ridiculous, but that's what happened. And when I got the laboratory notebooks, of course, it was all over, at least as far as I was concerned, because you can see here, this is what this particle looks like under a desk lamp. So this is a picture taken before a TEM experiment to show the palladium particles, and the student had left the holder for the transmission electron microscopy experiment under a normal desk lamp, and the particles degraded like this. Well, palladium melts at over 1500 degrees centigrade. I mean, a desk lamp should not cause the palladium to degrade. And then she wrote in the margin, all of our particles do this. So she knew. And then time went on. And a lot of people questioned my integrity. And I really, really suffered for this. But in 2008, when they published that paper I showed you earlier with the grids, etc., they collaborated with a fellow named Jim Diorio, very, very well-known scientist at Lawrence Berkeley Laboratory. So I contacted Diorio, and after many years, he finally agreed to work with me. And we did a few experiments. This is one that we did. We grew a hexagon, like they say. Actually, another important thing I should have mentioned is you don't need RNA to grow these hexagons. They're just simply a kind of crystalline material that falls out of solution and then degrades very rapidly. That's all they are. They are composed of the starting material, palladium dBA. So knowing that, we grew very large crystals, about 100 microns across. You see one here on the loop. And you see both before and after the experiment, it's kind of degraded. It's not a very good crystal structure, but the ORTEP here shows the two palladium atoms, and you can see that this crystal is palladium dBA. It's just the starting material. And this is the paper that I wrote with Dr. Diorio in 2013, and these are Diorio's data, these are our data. Again, what are we showing? We're showing that in the beam of the transmission electron microscope, these particles degrade. He's showing here, here's the particle at a short time, sometime later, little particles, tiny, tiny particles of palladium are forming, and so you can see a very, very poor diffraction pattern here from them. But initially, there's no diffraction pattern because this crystal, well, it isn't, it's an organic material that's kind of not highly crystalline. And in our data, what you can see is something similar. You can see that initially you see something that looks like a hexagon, and then you see these small pots. These are, again, the palladium that's left behind. We did so many experiments together. No one ever acknowledged this paper or showed it. Even after we published this paper, There were people who were denouncing me and saying that I'd done all of this because I was jealous of them and other kinds of reasons. It's unbelievable, really. So this is what they were saying. They were saying they'd made palladium. And that was a self-deception at the very least. And this is what was actually happening. What was happening was their starting material, shown here, was aggregating, or was forming a kind of crystalline aggregate or quasi-crystalline aggregate that then you could degrade in a beam, and then these central palladium atoms would coalesce into small particles. So we published 7 papers showing that they were wrong. People then later criticized me saying, why did you report them? You should have published papers. But I did publish papers. No one ever seemed to care. So the system is not working. When people do commit falsification, and I don't care whether this was intentional or not, it was falsification. And certainly once they had been shown the data, they should have known. And yet they refused to admit their mistake. If it was even a mistake. If it was a mistake to begin with, by the end it certainly was not. And my question is this: if I had not reported this, how long would it have taken for the truth to come out? As it was, it took 10 years. And as it is today, only one of the papers has ever been retracted that they published. And that is the paper in Science, and that's because the editors of Science were so ashamed, they retracted the paper. And in fact, despite the judgment of the US Congress, as you see here, letter from the Congress of the United States to my chancellor, this is the House Science Space Technology Committee, has been aware of a long-running case of research fraud, which was supported by a grant from the NSF, awarded to your university, and so on. I don't want to go into the details. This just shows you to what levels this rose. The Congressional Committee wrote to them. The National Science Foundation did a 10-year investigation, and on and on and on. So the editors of Science retracted the paper. But my university never corrected the record. They still haven't corrected the record. Instead, they started an investigation against me, saying that I had stolen the samples from those guys. Now, we had the same grant, and they knew it. How can you say someone stole samples if they're working under the same grant? And so I studied the law. I sued the university for retaliation under state law. There's actually now a federal statute that would have protected me, but unfortunately that federal statute was passed by Congress in 2018. Too late for me, sorry. So I used state law and I sued them. And in fact, during this time I started writing this book called University Responsibility for the Adjudication of Research Misconduct, The Science Bubble, which is what I see as a major problem. If it takes 10 years to correct such an obvious mistake, what is happening to our world of science? And in a sense, you could say that's why I'm here today, because as I began to see what was happening in my own university, I started to go overseas. And in fact, I spent a lot of time in Poznan with people who I could respect and work with, and I collaborated with them. And then I began running international programs because I couldn't justify to myself just leaving like that. I brought students with me. And in the end, since 2006, I've brought over 200 students from my university either to Poland or to China on different kinds of either study abroad, research experiences, whatever I can do. That's how I survived mentally, because it's very hard to maintain your composure and your balance when everyone around you is telling you you did some terrible thing. Even if you know you were right, it's very hard. So my conclusion, unfortunately, is that none of us are immune from this. Not me either. When I look back, I realize there are times when I too have deceived myself. Of course I have. We all have, actually. Sometimes, most of the time, it's not that serious. Most of the time, we correct it, we realize it, we do something about it. Curiosity and self-awareness can still resolve this. I'm convinced of that. But we have to teach that curiosity and that internal awareness to our students. And we have to go all the way up to the top and say, we need a different way of looking at science. This way of establishing merit, of making stars. Some people are superstars and other people are down here. That's ridiculous. I don't believe anyone is that good that they need a $50 million grant when other people can't even get enough money to support a graduate student. It doesn't make sense. But that's where our system has gone, unfortunately, at least in the United States. But of course, what happens in the United States seems to happen in many other places. So I'm very concerned about the trends that I see. Thank you very much for inviting me, and a special thank you to Ryszard Adamiak in Poznan, who was one of the people who really helped me so much through this. Bogdan Skalski, who's here in the audience, who also was there during those crucial years and really helped me and really saw this firsthand, and that was very important to me. And then graduate student Donovan Leonard, who I would say was the person who first came to me and pointed out to me these problems. And when he did, I listened to him, and I'm very glad I did, despite all of the things that I may have experienced that were not so pleasant. But I'm so happy to be here today, and I hope that all of you take away something from this that will help you in your careers, and hopefully the whole awareness of our community can begin to change. Thank you very much.
Thank you, Professor Franzen, for your talk. And actually, it was like a criminal movie, going through all that story. So not only science is very attracting, but also like meta science, what is going behind our laboratory table. And if there are some questions in the audience, if not now, so I will ask still Professor Ryszard Naskręcki to say some word. He is a host of our ECOTECH seminar. And still Professor Franzen is for a few days in Lublin here. So if somebody wants to talk about his story, her story, and about some feedback on this talk, so it's very welcome.
Thank you, Mr. Rector. So I'm very sorry that I'm late, but I have been in the City Hall. So today I had a short talk in City Hall concerning anyway to ECOTECH, and I want to promote ECOTECH. In my opinion, it was also important to be there. So I'm very glad that our ECOTECH-COMPLEX Seminar is growing, that we have such prominent lecturer as Professor Stefan Franzen, which is a prominent scientist, very famous scientist, but also our good friend. And I hope that Professor Franzen will find also the direction to Lublin, not only for this seminar, but maybe also for the future. I don't know, we have some tens of people in the internet. It is possible to ask them maybe for some question or not much. Do we have such information? No. Okay. So thank you again. Thank you, rectors, for your participation. Thank you, my colleague from Poznan, but thank you all the people which are here in this auditorium, but also in the internet. We will continue this subject because, as said Professor Gruszecki, this is a very important subject, the question especially for the young people, for the PhD student, for the student on the level, even the level of master, to see how difficult it is to say what is true in science. It is not very clear, and it is not a very simple way to say it is absolutely clear, and we should be very careful about this. So thank you very much for Professor Franzen, for all of you here, and I hope that even in December we'll have the next seminar. If not, I invite you for the next seminar in the last Monday in January. Thank you very much. Wonderful. Thank you, Stefan.